Welcome friends, this is Ibad and I'm here with another video from Ibad's Biology. We have been talking about various cellular organelles previously and today we are going to discuss another important cellular organelle that is Golgi apparatus. So let's start. What is Golgi apparatus? Golgi apparatus is a membrane bounded organelle found in eukaryotic cells and it is made of a series of flattened membrane bounded sacs known as cisternae. So actually Golgi apparatus is a bundle is a stack of membrane bounded flattened sacs known as cisternae. So we can call cisternae as the simplest units of Golgi apparatus. Okay. Golgi apparatus was discovered by Camillo Golgi in 1898 and that is why they were named as Golgi apparatus or Golgi bodies or Golgi complexes. Cisternae associated with vesicles and tubules form the Golgi apparatus. So cisternae are not alone in Golgi apparatus. They are associated with vesicles and tubules. Okay. These sacs, these cisternae are about 1 micrometer in diameter and they are bounded by a membrane and enclosing a space known as lumen. So as we now know that Golgi apparatus is a stack of cisternae, a stack of membrane bounded sacs. Each stack of cisternae or each bundle of cisternae is also known as dictyosome. And all the dictyosomes of a cell constitute complete Golgi apparatus. So we can call that dictyosomes are units of Golgi apparatus. Okay. Each dictyosome may contain 5 to 6 cisternae or in some cases they may contain up to 20 cisternae. A collection of associated vesicles and tubules surround the dictyosome and that tubules and vesicles they give a lace-like appearance to the peripheral area to the peripheral region of dictyosome. As you can see in this diagram, this is Golgi apparatus and we can call this complete bundle a dictyosome. This is a stack of these vesicles. Cisternae. Okay. So these are membrane bounded cisternae enclosing a space known as lumen. This is lumen space or cisternal space. And these spherical structures, these are vesicles which are associated with these cisternae along with these tubules. Okay. So this complete structure, this represent a dictyosome composed of cisternae enclosing a lumen, vesicles and tubules. Okay. Golgi apparatus has 3 compartments: the Cis Face, Medial region and Transphase. Cis Face is also known as proximal face or forming face and the cisternae in this region they are nearest the endoplasmic reticulum or towards the nucleus. The medial region is composed of central layers of cisternae while the transphase, which is also known as distal phase or maturing phase, this phase is composed of cisternae farthest from endoplasmic reticulum. As we will discuss later that the main function of Golgi apparatus is modification and packing of secretory products like proteins and lipids. So the proteins and lipids they are received at Cis phase and the matured and modified proteins are released at the transphase. Okay. In this diagram you can see that this region is known as Cis face and this Cis face is towards the endoplasmic reticulum. Endoplasmic reticulum is somewhere here. Okay. This region, these layers of cisternae, this is known as medial region while this phase is known as transphase. Okay. You can see that the incoming transport vesicles come and join the Cis phase and the matured vesicles they bud off from the transphase. Proteins and lipids when they are received at Cis phase in incoming transport vesicles, the vesicles fuses with the cis membrane and the contents of vesicles are released into the lumen of cisternae. where the enzymes are there for modifications. Okay, so as the protein and lipids progress from cis phase towards the trans phase, they are modified into functional molecules packed in vesicles and are delivered to intracellular or extracellular targets. Okay, so along with modifications of proteins, they are also tagged for their proper destination. In this diagram, you can see a clear picture of how Golgi apparatus works. So Golgi apparatus work in collaboration with endoplasmic reticulum. You can see this is endoplasmic reticulum. Okay, and this is Golgi apparatus. So this phase of Golgi apparatus is known as Cis Face towards the endoplasmic reticulum. And this Cis Face receives vesicles from endoplasmic reticulum. These vesicles, they come from endoplasmic reticulum and fuses with the Cis Face of Golgi apparatus. The contents of these vesicles are delivered in the cisternae, in the lumen of cisternae, and the contents, the lipids and proteins, while they progress from cis to trans phase in this direction, they are modified. Okay, and finally the functional and modified products, they bud off from the trans phase. This phase is known as transphase, and this transphase produce vesicles containing mature and modified products, either lipids or proteins or some other secretory products. So along with modifications, these vesicles containing proteins, they are also tagged for proper cellular destination. They also have the address attached that either this protein or this lipid will become part of cell membrane or endoplasmic reticulum membrane or other cellular parts, or they may be secretory proteins which are secreted outside the cell, exported outside the cell, or some of these vesicles may become lysosome. Okay, so along with modifications, these proteins are sorted and transferred to proper targets. Now let's have a quick look at the functions of Golgi apparatus. As we already mentioned, that the main functions of Golgi apparatus are modifications, packing and transport of secretory products like proteins and lipids. Golgi apparatus are sometimes referred to as a traffic police of the cell because they play a key role in sorting of proteins or other membrane constituents and directing them to their proper destination. Okay, they transport different components like proteins and lipids to their proper targets, either extracellular or intracellular. They also play a key role in the synthesis of polysaccharides from simple sugars and combining those polysaccharides with proteins and lipids to synthesize glycoproteins and glycolipids. Okay, so the synthesis of polysaccharides and the synthesis of glycoproteins and glycolipids from those polysaccharides, it is also the function of Golgi apparatus. Lysosomes are also formed from Golgi apparatus, so the formation of lysosome, and some secretory proteins are carried in vesicles and they are exported outside the cell by exocytosis. Okay, so this was all about Golgi apparatus and their functions.